before we start. Okay, here's what's happening tonight. Okay, we got a little bit of a late start, but we can do this. I'll keep track of the time. We're going to get through this introduction to what galls are, what are plant galls, and then we're going to have a bit of a break. We can have video, and I think that's where we'll actually have Joyce's video start, her YouTube, and then we're going to have all biology, how it works, what's happening, and then another break. And so as you see, there'll be perfect times if you save up your questions. If you absolutely can't wait, write them down, don't forget them, put them in the chat, and Kim will save them. But I've given us plenty of time for questions, and we can go afterwards with lots of questions too, although Kim may cut us off when we get past our Zoom time. We'll see. Okay? So let's go. Okay, got it all in mind. This is what's happening. What's a plant gall? Well, here are some. These are leaf galls. Oh, there's a stem gall, and here's another leaf gall. Okay, these are all Joyce's photos, so I have to say the good part of this talk is all her. The rest of it is me. Okay. Well, plant galls are actually made of plant tissue. But a gall is plant tissue that's not a normal plant organ. It's usually induced by another living organism. In fact, it is induced by a living organism. And this is where we'll look at different kinds of plant galls. But mostly we're going to be arthropod plant galls tonight. Host plant. Okay, we need to know that. You have to pay attention. You need to figure out what you collected your gall on, or we'll never be able to help you figure out what it is, even if we can ID the insect type, because so many varieties, especially, and the thing with the insects, we have different ages of insects. So if it's larval, it's going to be really hard for anybody to tell you what it is. So again, the plant grows totally plant tissue, all in response to a chemical signal from the gall-inducing, in this case, arthropod. Ceccidology is the study of galls, and we actually started it with Malpighi way back in 1679. Some well-known gall biologists include the late Alfred Kinsey. So here's an example. This huge thing, and I wish I had Joyce's ability to circle it, but you can actually look on at the middle of this picture, giant burly thing from, that's from a mistletoe, okay? That's over several years, this tree was responding to having the mistletoe roots cut down in its circulation. So it responded by, as it would to several, if you were to hammer on it in the same place for a lot of time, it would do the same kind of response. So, is it a gall? No, we say it's not, because it's trauma, continued trauma from the mistletoe attachment, but it doesn't always cause a gall growth, and mistletoe does not benefit from this gall growth. So mistletoe doesn't get any good out of it, and it's just continued trauma that causes this strange shape. So we're looking today at arthropod-induced galls, and these do not include many of the galls caused by fungi. There are gazillions of them, or yeast. We're not going to talk about those. There are galls that are caused by plants. We're not going to talk about those. And there are galls that are caused by bacteria. Classic crown gall. Okay, most people who have studied biology have seen crown gall. Okay, but we're not going to look at that either. We're going to be very narrow-minded. Remember, my specialty in entomology is cynipid gall wasps. So that's really the stuff I'm interested in, as you will see in tonight's talk. Okay, so arthropod-induced galls. You recognize manzanita? This is caused by an aphid. Okay, these are galls caused by mites. These are galls caused by insects, orders, thrips, bugs, Hemiptera, beetles, Coleoptera, moths, Lepidoptera, flies, Diptera, and wasps, Hymenoptera. All of these are arthropod-induced galls. So how do you describe a gall? Well, you start with what is the host plant? You do need to get me an identification of that, or I won't be able to begin to tell you what the gall is. We need to know what the host plant is. We need to look at what part of plant it's growing on. We'll be reviewing this later in the talk. So whether it's a leaf gall, petiole gall, stem gall, bud gall, flower gall, and root gall. How complex is the gall? Is it a simple pouch gall? Most of the hemipterans and aphids make really simple pouch galls. Or does it have complex tissue layers? You'll see some flies and lots of wasps make very complex galls. Okay, how many of the galling arthropods are inside any given gall? Some galls are monothalmous, one and only one. Others are polythalmous, many, many, many tiny gall inducers growing inside the same gall. The other thing we need to look at is the shape, the color, and other specific details of the gall. These are usually specific to the arthropod gall inducer. So we need to know all this before we can begin to tell you what you've got. We will have to name it, and this is important to back up. I will have to name it for the gall-inducing arthropod, that is insect or mite, that causes the gall, and not the gall itself. Once upon a time, we had people describing galls. It's not allowed in the international rules of zoological nomenclature. There you go. So you have to name it for the living organism and not the gall. Okay, mites cause galls on willows. They actually have very similar to the leaves like this on many leaves of many different host plants. So these are mite galls. They're kind of blistery galls and rather simplistic. Here's an interesting one. These are thrips galls. And these are thrips on myoporum leaves. And I told you I wouldn't talk about dissections until later, but I've got this gorgeous one that Joyce did. This is the thrips inside that gall you just looked at. He's very, very tiny. Think 1 to 2, well, maybe 3 millimeters long at most. So there you go, thrips gall. Often you get roll and pouch galls. Here's 2 galls caused by aphids. And beetles can cause interesting, now they might be simple stem gall swellings like this weevil gall, or they can be a little bit more complex. Moths can be very interesting and cause many different kinds of galls. So there you go. This is a moth gall on, it's a stem gall on Baccharis coyote bush. Okay, flies in many different families form galls. Okay, we've got tephritid fruit flies that make these interesting goldenrod galls that are widely studied in the East because they're very, very common, easy to find, and a biologist's dream. You can do all kinds of studies with them. So, or you can look at this simple midge gall. This looks a little bit like some of our cynipid galls. In fact, Joyce's and my friend, colleague at UC Berkeley, Diane and I described a fossil gall that looks just like this one, which is a willow tube gall on willow. And it was on an oak leaf, and we said, well, we're going to call it a cynipid, but we don't know. We can't prove it. Because when it's fossilized in rock, we just don't see what's inside. Okay, wasps in several different parasitic families, including Cynipidae, and wasps in the Tenthrodinidae group, which is very primitive wasps called sawflies, form galls of various complexity. Some of them have internal chambers. And here is a cynipid gall, and that's a rose gall. And here is a willow sawfly gall. So, Okay, galling arthropods manipulate plant growth by sending chemical messages to turn on particular plant genes. They arrange for continued access to nutritive tissues while their larvae are growing. So these form an amazing variety of shapes, colors, all from plant tissue. Here's a cottonwood petiole gall, cottonwood leaf aphid gall, willow tooth midge gall, a willow tube midge gall. Here's another sawfly gall on willow. The willow beaked midge gall midge, okay. Here's a willow rosette midge gall. Here's a cat claw leaflet midge gall. Here's the sagebrush sponge gall midge. Okay, again, all these are fly galls caused by Cecidomyidae, and we'll get to that in a little while. Here's a woolly stem gall midge, okay, the gall caused by a midge, and a saltbush pom-pom midge gall. It's a bud gall. You can see a little bit different area of the plant. Coyote brush stem gall, also a midge, and a coyote brush bud gall caused by a midge. Here's a Ceanothus vein gall caused by a midge, and a Ceanothus vein leaf, also the gall is caused by a midge. So these are all done by all flies in one family, Cecidomyidae. We'll talk about those soon. Okay, here's another rabbit brush cotton gall midge. And yes, no? Okay. I thought I heard Joyce say something and she would stop me if I said something wrong. Okay, here's a spiny cone gall midge. And at first I'm like, cone, I was thinking cone shape, but this is like a pine cone. Okay, juniper urn gall midge, juniper gall midge. Here's a creosote bush gall midge. You notice all these are really colorful and every one is different and distinct. If you haven't noticed, I'm sitting in front of a whole poster of different, these are all oak galls, actually caused by oak, cynipids in the, that I'm in front of, in the family Cynipidae, in this tribe Cynipini. So we'll get to those in a minute, but those will be last because that's my favorite. I save them. Okay, here's another creosote stem gall midge, also Cecidomyidae. Here's another bush paddle gall midge. So lots of color, lots of variety. Okay, now here's the cone gall midge again. This one's a little older than the last one we saw, but this is on creosote. Sage leaf gall midge. And now we're getting to my favorite group. Now we're getting to the gall wasps in the group Cynipidae. This is a chinkapin flower gall wasp. And you notice that you see this is on the flower, but this huge round gall wasp, or gall, is forming on the flowers. This is a leaf blister, also caused by a cynipid. This one is caused, chinkapin were the last two, it's a little shrub. This leafy bract is on Rosa, wild rose, and this is also caused by the gall wasp. Nebulosa, rose gall wasp. And these are one of my favorites. And this is a spiny leaf rose gall wasp. And notice the little spines, they get very strong and sharp, probably to discourage grazers who might otherwise eat the rose leaves. Rose pincushion gall wasp. This is actually an alien invasive species. And the species of wild rosa it's on is equally alien invasive. This is a Linnaean species, originally described by Linnaeus in Europe because it came from Europe. Here's a rose blister gall wasp that actually is a native Californian on our native California rosa. All right, now we're on to my favorite. These are all caused by cynipids in the tribe Cynipini, oak gallers, all limited to oaks. Okay, look first at the part of the plant that's galled. So here's a wasp gall on a valley oak leaf. And I really like this one. This is a Confertus gall. We'll get back to this. This is one of my very favorites. This wasp bud gall on scrub oak is actually, was on the cover or something like this. This is Joyce's picture, but a very similar picture was on the cover of Ron Russo's original book in 1981, I think, whenever it came out, the California galls, Plant Galls of California, I think it was called. All right, here's a twig gall on blue oak. And this one is actually looks a little like coral. And in fact, it's called Corallina is the species name. It's very orange this time of year. And here's a flower gall. This was actually on Coast Live Oak. This one occurs is actually purpley in March, which is when the oaks are actually flowering. Okay, here's a stem gall, the last in the series, looking at the parts of plants. This stem gall is on canyon oak. All right, so basically, now that we're getting into the Cynipede and Cynipede in specific, we're looking at gall wasp. We see complex galls, multiple tissue layers, not simple pouch or roll and fold. There's always a special internal chamber for the individual larva. If it's a polythalamus gall, there'll be lots of chambers. If it's a monothalamus gall, there'll be just one. We'll note, and we'll get into this later, that alternate generations produce different galls on the same or different host tissues. And finally, we're looking at the gall that can actually, the same species can gall alternate oak species within the same oak clade. So there's several Quercus species which we classify as white oak. And I'll tell you, these cynipid wasps are smarter than me, at least as a botanist. They can recognize white oak and tell it apart from black oak. So they can you can find galls that you normally would find on Lobata on other white oaks, for instance, on blue oak or scrub oak. And I see them also on Oregon oak. So all kinds of white oaks will be able to make the same gall species. We think that, in fact, well, many ceciologists think that possibly new species develop when a white oak galler develops the ability to gall on a black oak or a canyon oak. That switching species in a major way actually causes or leads to new speciation. That's one of the theories at this time. Okay, black oak is another group. We'll see. Black oak includes live oaks, not canyon live oak, all the other live oaks. Okay, coast live oak, interior live oak, Kellogg oak, other kinds of black oaks. And then last of all comes canyon oak. These include the huge, beautiful canyon oaks that you probably see in your area, but also the teeny tiny, at most 2 feet tall, up in the high Sierras, huckleberry oak. And all of these can make the same gall. So we'll see the same suite of galls and the same species of gall wasp making galls on all of the species within this clade. Okay, the fall galls are the showiest. They have to overwinter, and here's one that's my favorite, Andricus fulawei. We're looking at Quercus lobata galls. I'll just go through this quickly because I want to give us all somewhat of a break at some point tonight. Okay, Confertus, also a leaf gall. This is multiple chambers. So this is a polythalamus gall. Each one of these is going to have a separate, or we could say it's a gall made up of a bunch of separate chambers. So here's a monothalamus gall. This is the club gall. Here's a kind of Okay, would you call this a floral rosette gall on lobata? And this is the jumping gall, which you may have seen or definitely will by the end of this evening. See pictures of galls jumping, which is a lot of fun. Okay, and now we're finished with Quercus lobata, the valley oak, and I've gone on to the blue oak. Here is a close relative of the star gall, but this one is sea urchin gall on blue oak. And here's Androcus bruneus, also on blue oak. And this beautiful one, Discalcaspis corallina, is also on blue oak. Lyodora pattersoni on blue oak. And now we're getting to scrub oak, and I think this is the only scrub oak picture I have, but here's my favorite, plumbella, beautiful, colorful plumbella gall. Now we're into the black oak group, and here is a root gall, which actually came up in my former boss's yard. Her oak tree had these in its roots. And Joyce and I had great fun with, Joyce cut this up dissected it for the photograph, and I got to ID them and point a lot of them for our records in the museum. All right, this is another one also on the same species, coast live oak. And here's another on coast live oak. This is used to be called bicornus because of its shape. It's a leaf gall. All right, now we're into the canyon oak group, and this one is Heteroicus sancticlari. I think of it as a kind of, I don't know, elf hat or mushroom gall. And here's another one, beautiful, also in the same group, a leaf gall, Lasius. And last of all, another leaf gall on Chrysolipus, which is also the canyon oak. And last of all, this might not be on Chrysolipus, although it looks like it is. This one is Heteroicus pacificus, a spindle gall. And yes, it's on Chrysolipus. All right, so we finished the first section, and I'm going to stop sharing. And let me see what yes, stop. Oh, no, I don't want to do it.
I think you're good. We do have a couple of questions for you. The first one was, how do the wasps deal with hybrid oaks? If they're in the same clade, do they infect as usual?
Yeah, well, they seem to handle it pretty well. And who knows, that could be some of the speciation, because oaks, as you know, are wind pollinated. And as some plant biologists have called them promiscuous, yes, they hybridize easily. And, but apparently the gall wasp recognize the clade, but they can't go beyond that, or mostly they don't.
Thank you. And then the second question was, do we know what characteristics attract wasp species to a particular oak?
Okay, I can only say from watching their behavior, and I'm only guessing, but when you watch a wasp move around, and actually Joyce had one yesterday, may have one again tomorrow, a live little, that was an inquilin, but she was doing typical behavior, okay? Palpating with the antennae, which is sort of tasting and smelling, because that's what they do. They're chemoreceptors in the antennae, moving along, so they're obviously smelling it and feeling it, right? And possibly, in this case, since she's an inquilin, she'll be looking for existing galls and looking for larvae, although we don't know, inquilins have different reasons. So, but mostly that's what attracts them to oak trees, I think, is by scent as much as anything else. But okay, we don't really know. I have to admit that when we get into what causes the gall, cytogenesis is, well, not yet enough studied. No definitive answers, just a lot of great ideas.
And the last question that is in the chat is, how how far do the wasps travel?
That is an excellent question. Okay. I don't know how many of you know about aerial plankton. Okay, really light, small insects, and most of these wasps are at least less than 5 and mostly less than 3 millimeters in length. Sometimes get swept up high in wind currents and travel all over. So some of them make amazing trips. Most of them don't. Okay. Most of them, but I'm always amazed at how far they can travel just on the wind. I've tried following individual wasps, the ones I thought I could follow, and they fly off beyond where I can see them. And sadly, since they're small, and it's very hard to radio collar these things. In fact, I haven't even tried. So just say they get a long way. I don't know. I can't. That would be a fascinating study, and we do want to try it sometime. So, but it is amazing that they get a long ways. And yes, you find them in the aerial plankton. That's a little scary. You can find them at the poles on ice where they won't do much good, but they're up there. So if there were any tiny oak trees up there, they would be able to gall them. Okay, any other questions? Okay, let me just see what I promised you guys. How's my time doing? I better check what I told you. The first one was, well, do we have any more time, Kim, for a video?
Sure. Did you want me to share that, the one that Joyce sent?
Yeah, I think that's okay. Is that okay with you, Joyce? Okay, we could save it for tomorrow or share it now. I think now would be good. Okay. And I'll take a drink of tea while you do that. You can see how difficult it would be to identify these guys. They look like little blobs. They look like late instar larvae also, and probably will be pupating soon. They look large enough.
So this is in a gall that's supposed to be monothylamous. So some of these are probably inquilins.
Well, that's even more exciting. So they might be inquilins. Yes, well, and again, you don't need to know that term yet. It will all be displayed in due time, and that's our next project. Okay, so this time I will be doing the screen sharing, and I will share it with, let's see which one. We're doing Gall Biology 2, right? Sorry, guys. Can't read my own screen as well as I'd like. But here we go, slideshow, and now. Here we go. All right, thanks for your patience. Okay, I'm going to tell you what we know about cecidogenesis. Cecidogenesis is how you generate a gall. And I'm going to give you 2 examples to start with, because these are both Hymenoptera. And while the Tenthrodinids, all right, I shouldn't say this, but I think their galls are a little less complex and a little less spectacular than the cynipids, but I may be a little biased. Okay, So the tenthrodinids are also Hymenoptera, and yes, their galls are interesting. Mostly, we'll find a lot of tenthrodinid galls on willow. We find a lot of cynipid galls on oak, and that's to begin with, all right. But this much we know, and this much I can tell you. We're not sure exactly what the chemical is, but at some point in the ovipositor, the female sawfly puts a fluid down made in her accessory gland, goes with the egg, and it causes the gall to start growing. So the gall will grow even if the egg dies, or if it closes and the larva dies. So that's the story. There apparently, whatever is causing the sawfly gall is actually deposited at the time of oviposition, when the egg is laid. Okay. Sawflies, if you don't know, have this long ovipositor, and often it's sort of jagged, like a saw edge. And they often deposit. Now this one is in a gall on leaves, but many sawflies actually live in wood, and that's what they need to be able to put their ovipositor deep in wood. So, all right, let's look at induction in gall wasps. Okay, in this case, the egg also comes down through the ovipositor, and it actually hatches before any gall growth happens. So the gall doesn't start to form until after the larva starts growing. We're not, okay, some of my friends who also study cynipids say that they're sure it's not a salivary or anything in the saliva. I don't know that that's so sure, but it does manage, whatever it is, the chemicals that induce gall growth are actually turning on plant, either counterfeit plant hormones to turn on plant genes, or they are deliberately or directly turning on the plant genes. So plants will form certain kind of growth. What's even more amazing is that plants will grow in a certain way, making a certain shape, and then the growing larva will give it a new command, a new chemical, and it will change, and will change color, it will change shape. There'll be another wall formed and another layer formed, another tissue layer formed. So it's very fascinating to watch the growth, but it doesn't happen without a larva. If the egg dies, no gall will form. If the larva dies, gall growth will stop. So if you see a deformed gall, often the larva died, and you can see why. Sometimes some of my big California oak galls, you'll notice they get damaged. Rain will come down and cause them to get some kind of a mold, and they die, and the gall stops growing. And you just see the big dead area, big black mold area. All right, so two different, okay. So now we know a little bit about gall induction, which is we don't know. And I have a friend who, she is a botanist who's working on gall induction at the plant hormone level. Other friends are working at different levels, but we don't have any definitive answer. My friend, the botanist who's working on induction, is actually working on tephridids. So her answer wouldn't be good for me, because tephridid flies are not cynipid wasps. All right, so anyway, two families of insects are found mainly in galls. The Cecidomyidae gall midges, we have lots of species, lots of plants, and many are undescribed. However, the more that they've done work on the gall midges, we now have a whole group of wasps in there, not wasps, flies in there, that are not in galls, that are not connected with galls in any way. So we can't say all gall midges are in galls. We used to say they were, but now we don't. On the other hand, all gall wasps are still and always found only in gall tissue. Not all of them are the gall inducer, and we'll talk about that much later. But basically, cyanipids are found only in galls, and the original gall midges were found only in galls, but now the family's enlarged and that's not true. Okay, now we need to look at gall midges because we have lots of them. And Joyce is very good about knowing them and has taken many beautiful pictures, and I need to learn more about them. So that's my next challenge in life. When we can get out more, I'm going to go collect more and study them. Okay, here's one on honeysuckle. Beautiful midge. You can see she looks a bit, okay, they're a primitive kind of fly, long antennae, small heads, look a lot like mosquitoes. That's a gall midge. Midge is a mosquito-y shape, long-legged. Okay, here's another gall midge. This was on coyote bush. This one is on Ceanothus. Beautiful gall, complex looking. All right, now we're gonna look at the gall wasps. And I promised I'd tell you about this, whether you wanna learn a new term or not. Okay, close your mind. If you're getting a headache, close your eyes. But heterogeny, it's when you have alternative generations. We have this kind of process happening in aphids. We're not going to worry about aphids today. We are going to worry about gall wasps because in cyanipids, the alternate generations, which is heterogenes, you have a one generation and then a different kind of generation. The alternate generations actually can produce different kind of galls and often on different host tissues. And in Europe, on different host plant species, which is really interesting. Okay. Heterogeny, alternating generations. So before I get into this, I'm going to tell you, I don't have it listed, and it may come up soon. We have a truly asexual generation, okay? All female, all of them ready to lay their 300 eggs or 350 eggs, depending on how many they have, ready to lay them the minute they come out of the gall. And then the alternate generation is bisexual, both males and females, usually in about equal numbers. And they're ready to mate, and then only females will be laying eggs. They'll be and it could be a totally different kind of gall and on a different tissue. So here's Anthracus philowei. And this one is a beautiful fuzzy gall, leaf gall on Quercus labata. And Anthracus confertus on Quercus labata and Clavuloides on Quercus labata. These are all fall galls. We do not know the alternate generations of this group. Wiltsea, we don't know either. So I'm getting into the more details. We'll get into the heterogeny in a minute, specifically. Okay, here's jumping galls. And I think I showed you some of this before. And Quercus echinus. Okay, so these are ones, oh, I guess it's duplicated in this section, sorry. Okay, Pattersoni, the root galls. Sorry, I didn't realize I had that here. I took it from another section, different talk. Okay, so here we are. This was supposed to be the next slide. Bisexual generations followed by alternate unisexual generations that are all female. We can see these in bug galls, like grape phylloxera. We can see these in oak gall. Okay. I told you we weren't going to talk about aphids, but we're going to talk about their relatives only because they have fascinating alternations. In the case of the Cynipini, these are the oak gall wasps, we have only 2 generations a year, but we generally have the pattern of 2 different generations a year. One goes through the hard, long winter time, and usually that's the unisexual generation. everyone that comes out in the spring will be ready to lay eggs. Then in the spring, when there's lots of plant tissue and warm weather for growth, that's when we'll get the bisexual generation coming out, mating, and get the variety we need. We need genetic variety, or no species lasts over time in evolutionary terms without some variety. So that's where we get the variety, but we get that only with the alternating generation in the spring. It's a short-lived generation, and often the galls are almost hidden, very, very hard to find, and they don't last long. So here is, okay, you've seen the gall making in insects. If you haven't read it, don't worry about it. Okay, this is grape phylloxera. This different pattern, okay, we see some happening. Let's see if I can do the pointer. Okay, we see them happening in the root galls, then we see them up here and okay, happening higher up at different kinds of galls at different times of the year. We've lost it all. By the time this species gets over to California, we've lost the alternate generations. We have only the female root gallers. So don't go out in your vineyards trying to dig up all this. You'll just get the female root gallers. Yes, we do get phylloxera, and the only way to handle it is to get root stock that is impervious to phylloxera. All right, but this is the cynipid. Oh, we've taken this actually from a picture, not a very good drawing, and I've adapted it. Okay, what we will see is that they're not always the same. Okay, the females don't look the same, and this one that I adapted it from is an eastern gall, but I've adapted it to our western species. So we've put it on Cynips douglasii, the star gall. This is a pink star, and we'll show you that's the one. We call it spiny turban, is what you'll see in Ron Russo's book. It looks sort of star-shaped. It's beautiful, and it's bright and pink. Okay, these are Bonophthalmus. Okay, all of these are on leaves now. They'll fall to the ground and in very early spring, and this will be, probably in about February, March, they'll start coming out of the gall. So in February, March, the females will come out of this gall, and they'll lay their eggs into a bud. And here's the bud gall. Okay, Joyce has this beautiful one where she's actually dissected the bud, and you can see the next generation of gall wasp. This one happens to be female, but there were males too. So there'll be both females and males. They'll have to mate before they lay their eggs. They'll lay their eggs on leaf galls. So the bud gall lasts just through April. And then by late April, we've got the mating happening and leaves getting the eggs are being laid in leaves. And by June, July, we've got these things forming, and they're obvious. That's the cycle. Okay, both males and females come out in the late spring, but we have females only coming out in January and February. All right, so here is one on your coast live oak, and this is Quercus palmiformis, which is sort of another apple word. I think that's what it means. But this is the unisexual generation. So this is fairly large. It's probably what, 1.5 to 2 inches or more in diameter. And I've collected some just this last 2 weeks ago, actually. All right, this is the spring generation. Okay, these are soft galls. They don't last very long, and they dry up and they don't look like much. So you just see sort of brownish tissue from earlier years. But this is the alternate generation, and it's on the same oak species, but it's on a leaf And here, the other one was a stem gall. Here's another stem gall. This, the unisexual generation, is a stem gall, a spindle gall. And we're going to see this again because these actually have an interesting inquilin story. Okay, the spindle gall is unisexual. All of the females come out. And here we've got the sexual generation on leaves. So the spindle gall was a stem gall, and now we've got leaf galls as a short sexual generation. This one is the unisexual generation on blue oak, and it's also a single wasp in there. And here is the sexual generation, also on blue oak. Not very long lived, turn brown later, won't look like much. So, okay, here's one that really disappointed my friend Bob Zuparco when I told him. He'd been collecting these in pictures. He mostly looks at the parasitoids of them. And he was like, okay, what do we call it? What's its alternate generation? And when I told him the bad news, because this one is bicornus, which he thought that at least describes the shape. But the alternate generation is a flower gall, and it's called dubiosus. This happens to be a male, so you know it's bisexual. Obviously, they're both males and females coming out of these. He's like, why give it a dubious name? We should call it bicornus. But the rules of zoological nomenclature was whichever species was described first. So that's why we call it dubiosus. He beat whoever named bicornus. All right, so we're going to stop the screen sharing. And okay, time, how are we doing time-wise? Do we have time, do you think, for questions?
We do have a couple of questions.
How do I tell the sex? Oh boy. How do I recognize the sex? Well, first of all, unlike their counterparts in the vertebrate world, male cynipid wasps usually aren't going to hang their genitalia, Ediegus is the name of it, out. So it's going to be inside, so you'd have to dissect the thing to figure out whether it had one. Okay, so I'm not looking for genitals, but these guys are so cool. They will actually give it away in their antennae. Okay, males have super long antennae, and let me see if I can go back to that last picture. Okay, I'd have to screen share. Do you want me to screen share for a second? Let's see, how did I tell he was a male? Quick screen sharing, if I can figure out how to do it. Yeah, and okay, this guy is a male because he has very long antennae, and most importantly, he has a crooked section right here, and it's this crooked segment of the antenna. Not just that they're long, but they're not club shaped. The females are club shaped. You might notice that when we look at more wasps. So the males have longer, skinnier antennae, and they have a little notch in their third antennal segment. The other thing I notice is his gaster, and while he's not hanging his Ediegus out, the females have a wider gaster. We don't call this the, abdomen, because it's actually only the latter section. The first section of the abdomen is actually as part of the mesosoma, which would be thorax in insects. Okay, it's actually, the wasp has glued on the first antennal segment to its thorax. So we have to give it funny names. So I'm calling it a gaster for short, but I could call it metasoma, whatever. All right, this guy is small and narrow. Females tend to be larger, and they also do show, female is the one that's going to show a piece of an ovipositor. And that's interesting because the male, you know, we look for males' genitalia hanging out and not female genitalia hanging out when we look at vertebrates as a rule. Just a little different. But mostly I look at the antennae.
Okay, and then another question was, when the all-female generation overwinters, is it as a larva inside the gall?
Well, I think it's actually already mature. And I can show you more of this when I do my videos for you tomorrow. Because I've tried, I can't seem, I had a little microscope device, but I can't get software to work with it, so it's okay. My college said, don't do it, get something else. Well, yeah, right. And they tell me, oh yeah, you can invest some money and buy your own. I'm like, no thanks. But anyway, I will give you videos, so you will be able to see it. You can actually check, and if you cut up most of the galls right now, at least half, if not most of the Androcus kingi will already have finished pupating and will see an adult just waiting to overwinter. So in most cases, I think they overwinter as adults or just almost adults, late pupae. No, I think they're finished pupating as a rule. And that kind of fits. I mean, okay, you can look at even the jumping galls. Once they start pupating, they stop jumping and they wait months and months and months until next March. And then they come out and start the cycle again. So that's the best guess, is that majority of them, at least all the ones I know of, inside the gall.
We should see some tomorrow.
Yes, and thank you. We will see some, I promise. Between Joyce and me, we will have some.
So another question is, how do scientists figure out how species induce galls?
Well, this is really amazing. And this mostly, okay, the person I've spoken to most about gall induction is my friend Carol Mapes, who is one of the Pennsylvania State Universities. And she is the botanist who works with tephridids. These are picture wing flies, or we would call fruit flies, that make those big galls on goldenrod. She says that the amount of chemical, and these are plant hormones, in order to get to tell how much plant hormone is being used by the plant, you actually have to wash it with an extreme amount. Now, I don't really understand it, but apparently the amounts are, when their amounts are that small, you have to over wash it to see if that's what's affecting it, or to see how it's affecting it. So she's actually done tests for the amount of plant hormone present in her galls at different times. And that's been most of her work. So that's the best I know. Let's see, as far as induction, the early studies for gall wasps, and this is really interesting, way back in 1915, they ground up gall larvae and injected that material into a leaf or a stem, and they got a gall, which is pretty amazing. Now, we haven't revisited those experiments, so there might be some problems with them, but we're working on that. That's another, down the road in my next life, or even just later in this life, I plan to do that. What would be really fun is if wasps can do it, can we inject the oak tree and have it grow us furniture, our house? Who knows? That would be very interesting study. Okay, anything else? All right. The other two units are fairly short. Do we want to, I think what we will do, okay, of you guys here, raise your hands. You don't need to turn off or turn on your microphones, but just raise your hands and wave them if you have seen our deep look video. If not, we'll show it next week. Some of you have, some of you haven't. Okay. I'm sorry, and oh, I'm really sad for those shaking your heads. Okay. That is on the website that Kim has up for her park, and I love it. You can actually click right on and all of the stuff going on, and you can watch the video. Highly recommend it. Joyce and I worked on that video together with KQED. So we are the proud producers. No, we're not the producers, but we were the scientific consultants for that video. They kept us quite busy. Okay, the one I will show later today, if we have time this evening, is the longer one, because this was the one we did, okay, even though it says it was more recent, we actually did it in 2004 and 2005 is when it came out to BBC. I will show you that one only because it illustrates some of the great things I'm going to tell you about in the next sections. So, ready to go?
You want me to show that one now?
No, we're going to show it next after. I want to talk about the next section. I'm sorry, guys. I know I'm long-winded. How many can't stand my talking anymore? If I see a lot of hands, I'll give you a break. Oh, all right. One more, and then we'll start. Okay, my throat is now moist enough to talk. All right. So we will go back to screen share. And now I have to find the next one, which is Gall ecology. And we were going to do the slideshow, and let's start from the beginning. Whoops. Right. So Gall ecology, the arthropod itself, the insect in this case, is actually a parasite on the host plant. I like to argue that my little parasite doesn't, oh, can you see me? Are we screen sharing? Yeah, okay. I like to argue that my parasites are not actually hurting the oak trees. I haven't seen serious damage from galls, but some has occasionally been reported. That's all I can say. But there's a whole ecology going on inside the gall as well. And can you hear me on this? Let's see. Can you hear me, or can you see it at all? You did?
Yep, we can see and hear it.
Oh, good. Okay. I couldn't see me, so I wasn't sure. All right. Thanks for your patience. I'm talking to an empty room now. Inquilins are the uninvited guests. And in this case, inquilins all belong to a tribe of Cynipidae. Okay, so we have a whole tribe of cynipids that are inquilins in the gall. They have forgotten or no longer bothered in the chemical that forms a new gall. So they don't or can't form their own gall. They move right into a gall that's already being formed. Now there are two patterns there, and we'll talk about them.
So Kathy, I think you need to share your screen again.
Oh, I didn't. Okay. I'm sorry.
You were before and then you stopped, so.
I'm sorry. Okay. Here we go. I apologize. I'm a little muddled, but here we are. Okay, I am now from current slide. Okay, I'm sharing now.
That's perfect.
Thank you so much. All right, talk over me if you need to. Let me know what's going on. All right, so anyway, we're gonna look at inquilins, the uninvited guests, and two patterns they form. We're gonna look at predators, which includes both birds and insects, and some of the damage they form, because even though it seems like being in your own little safe world, or as they say in our deep look, your own banana, You can have all the food you want and you're safe from the rest of the world. You're right out there, and predators can find you, and inquilins can find you, and so can parasitoids. And parasitoids are parasites that actually kill their host. Fascinating, scary, Halloween story for sure. Okay, so let's get going. Inquilins are uninvited guests. They eat the plant gall material. There's two basic types of inquilin behaviors. When the inquilins first hatch out, the larvae hatch out in the gall, some of them actually go and kill. They don't eat it, but they kill the gall wasp inducer larva. So the original Cynipene larva may be killed. They seem to be doing that in the case where the gall would soon be lignified and solid, and their larvae need it to stay young and fresh so they have food and tissue to eat. Whereas in other galls, they actually may change the shape of the gall. They may allow the gall inducer to live. And in that case, they can make the ultimate gall have very strange shapes. And that's what we're going to look at, the second type of inquilin. Not the murderer. This is the one that moves in and just does its own thing. Okay, without an inquilin here, this is a normal Heteroechus pacificus gall. It's a long, thin spindle. We've seen a picture of this before. This is typical on canyon oak. Spindle gall. This is what happens if it has an inquilins, because in this case, the inquilin wasp has laid her eggs all around the original gall inducer. So the gall inducer still develops in her own chamber, but each of the inquilins develops his or her own chamber. And those inquilins will cause the gall to get bigger and larger and have a very odd shape in the end, way distorted. So this has a synergous inquilin. We're not sure which species. And Joyce here has dissected a gall that had, I think this is Joyce, yeah, mine is much worse photograph. You can see in the center here, this was where the original gall inducer lived and ate, chewed through a hole and exited the gall. But here, all around it, surrounding it in a circle, are the inquilin chambers. So the inquilins developed, they were smaller than the gall inducer and they have smaller chambers, but they've all developed from the same gall. All right, here's the normal Heteroechus daisydactyly gall, also on canyon live oak. And here's what it looks like if there were inquilins in it. So it's changed not in shape, but in size and more interestingly, in the surface texture. And here's what happened when Joyce dissected these. You would see in the center here, there was a gall inducer. And look at all of the inquilin larvae, or inquilin adults, sorry. These aren't larval at all, these are adults. And it looks like we have both sexes represented. Okay, so all of these emerged live. Nobody died, they all lived through it. And here's a close-up so you can actually get a good look. Now, if you want to see how to sex these guys, okay, we want to look for this looks like a male to me. And if you look carefully, you should be able to see a notch on this long antennal segment here. And he's got a rather narrower gaster, whereas the others here have rounder gasters and different shaped antenna. The antennae of the females are a little bit club shaped, although these are not nearly so club shaped as fidgets. I'm getting confused there, but some of them are more club-shaped. So this is a male, this is a female. Very slightly longer, but mostly look for that notch in the third antennal segment. Okay, does that help? All right, so female, male, male, female. All right, so while a gall might seem fairly safe from predators and parasites, unfortunately, there are a lot of predators who do manage, because they know what's going on inside, they've figured it out, and they actually go after the resident arthropods. So a gall, because it's out there day and night in the same place, and unlike an active insect, can't fly away, the gall actually is maybe not such a safe place to have your brood live. Okay, birds often peck holes in the galls, and we'll see a lot of holes in those great big Californicus galls. They're usually drilled by flickers who love ants, but also apparently love wasp gall larvae. So they'll peck holes in the big galls with their long and hard beaks. Other birds, like our house finches, wait until the Andricus kingi galls are just finished. Well, they can tell, and I can't, but the Andricus kingi have just turned red, but they're not yet becoming beige and hard. They know that they're still larval and not yet pupal. And that's the point at which they knock down all the galls they find. And I don't dare walk on my sidewalk in bare feet, because with these hard broken bits of galls from when they've broken them out, they're pecking holes and trying to get out the insect. And they usually succeed, making a huge mess on sidewalks everywhere in my area. Okay, and sadly, even predatory insects await adults emerging from galls. So here is a predatory insect, one of my favorites. This is a snake fly. If you're a gardener and you see one of these in your garden, welcome it. Yes, it looks scary, but that long thing is not a stinger. That is an ovipositor. This female is going to lay eggs to have more snake flies. And if you've ever watched a snake fly eat up a bunch of aphids, you will know you want them in your garden. You want to attract them. Even though they look prehistoric and weird and scary, they're a lot of fun and they do a great job, better than any insecticide in your garden. Alas, they also like wasp larvae and wasps. All right, parasitoids. These are a special group of wasps with only a single host, as just like a parasite, okay? Think of fleas. All of those fleas live on one cat or dog, spend their whole lives on the cat or dog, taking little bits of the cat or dog blood and being happy. Well, a parasitoid has one host, but what it does is the parasitoid larva or larvae, we'll see sometimes they're multiple, they slowly devour the host insect. Usually parasitoids are larval or larval pupal, and they will go after the larva, which is still not specialized tissue, and slowly eat it from within. Now there's some really neat parasitoids that even go after things like ladybugs, but I don't have time to talk about those, fascinating as they are. But the parasitoid larva will slowly devour the gall insect until the insect dies. Okay. Most of the wasps that go after gall insects are in the superfamily Chalcidoidea, but there are lots and lots of families, and all of the chalcidoids have very long ovipositors and carefully lay their egg directly into the gall insect. Okay, their ovipositors have chemosensors, or you might say taste buds, okay? They can actually taste that this is not plant tissue, this is the insect. They lay their egg into the gall insect, and as their egg becomes larva and then develops slowly into an adult, it will slowly eat the entire gall insect larva. So in fact, there's a whole family of chalcidoids. These are called the Ormyridae, and I have one picture. Sadly, it's one I took. Joyce has got to get me a picture of an Ormyrid. So it's an old picture that I took of Ormyrids, and you can see how beautiful they are, but they're really, as I said, we could do a better job on it. They're all, metallic, beautiful, have fancy crenulations on their external tissue. So they're beautiful on the chitin on the outside. Okay, these are all parasitoids of only gall insects, but the Ormyrids go after not only gall wasps, they go after gall flies and a lot of other, maybe gall moths too. They're not that fussy, okay? So we'll see them on lots of different galls. All right, here's Pterimus californicus with a very long ovipositor. This ovipositor is reinforced with metal, and it must reach inside the oak apple gall. She lays the gall is already green and almost the size of an apple when she goes to lay her eggs. And that's why she has such a long ovipositor sheath, because it has to be brought all the way down, that drill bit. And it's actually reinforced with metals, especially zinc. So metallic reinforcement, which she's sequestered from plant food that she's eaten. Okay, by the way, she's a coppery red, but her mate, the male, is a metallic green. Okay, here's another wasp, also in the family Pterimidae, but she happens to be a metallic green. And again, you can tell this is female because of her ovipositor. Here's a male in the family Eurytoma californica. This is as big as the, bigger than the Pterimid you just saw, but as big as the Pterimus californicus we saw the first time through. These are as big as the larvae that are their host, and the larvae is the Androcus quercus californicus. So this is another one of those. These little eulophids, you can't see as well from this picture, but they're actually a metallic green if you look closely. You can see the green highlights. These are tiny, much tinier than the ones we just saw, and we're going to get a whole bunch of them inside each larva. So multiple parasitic wasps will feed on a single gall-inducing larva in this case. And here's the Ormyrid. An old picture I took, but you can see her beautiful metallic coloring and the sort of scalloping shape that she's got. Just gorgeous, beautiful sculpture on this wasp. So gall insects are stuck. They really can't escape. Here's another Pterimid. And this Pterimid is going to lay her egg into those poor, helpless gall insects. But they can recruit help. And we're going to see in this upcoming video and from my next picture that some galls will recruit ants to help them out. This is the honeydew gall, Discocaspis eldoradensis, on Quercus lobata, the valley oak. And it's drippy and sweet, and you can see the ant is actually feeding on this beautiful honeydew nectar. We call it honeydew. If there were an aphid inside, it would actually be, honeydew is the aphid's feces. They're liquid because it takes in a lot of liquid in drinking the plant juices, and it puts out a lot of liquid and a lot of sugars. So the ants will actually feed on aphid feces, which are called honeydew, but this stuff mimics aphid species or honeydew. So this is the Dishelcaspis eldoradensis, and it recruits ants. And we're going to watch that in this upcoming video. So let's see if we can get back to screen sharing, and I get off of share. Okay. Now it's, yeah, we still have time. Don't have much left, so we've got time for this. This is a full 5-minute video, but I love it. And it was the first one that I was involved in making, and I just think it was really exciting. My friends in Hungary did one section of it, and my friends, two friends in Britain, one in Scotland and one in England, did another section of it. However, yes, this is really fun. Listen hard, and I'm going to point out a few things I want you to notice.
This is the Attenborough one, correct?
Yeah. Now, before we start, I want 2 things I want you to look at carefully. Okay. He's going to show parasitoids, and you're going to recognize, oh, that's a pterymus or a pterymed, because it is, and it's a parasitoid. Now, he'll call them a burglar, but these are actually coming to feed on the gall wasp larvae, whereas that's in England. Now, when he's going to go to California. Well, he didn't actually make it, but I'll tell you that story later. But I brought California galls to Arizona where he was, and we filmed there. And then we filmed some here, and we kind of blended it all in. But I have 45 seconds in the middle of this 5 minutes. And then we go to Hungary, where he tastes honeydew from a honeydew gall there. And he sees another burglar, but in this case, look carefully. And now that you've seen enough cynipids, you'll recognize that it's an inquilin in the same family. So you look carefully. Now that's one. The second one you need to look for is right in the beginning. Okay, the very first wasp you see is going to lay her eggs. Look at her. She's shiny and black. Watch the females that emerge. They're larger, they're brown, and they're not shiny at all. They're hairy and kind of dusky looking. Notice that these are alternate generations of the very same species. So the females from one generation to another can be very different. He doesn't point it out, but you can see it. Okay, thanks for waiting.
To provide a home where before there was none. But some insects have the ability to force a plant to do just that. They're called gall makers, and this oak tree is infested with them. This odd wrinkled object at the base of an acorn is known as a knopper gall. Inside, there's the tiny grub of a minute wasp. To understand how it got there, we have to go back to last spring. This tiny insect, scarcely bigger than a mosquito, is one of these gall wasps. There are lots of them flying around the oak flowers. Most of the flowers by now have been pollinated and are about to develop into acorns. The gall wasps too have mated, and this female is looking for a place to lay her eggs. She thrusts her ovipositor into the base of the fertilized flower and injects an egg. And that triggers a profound genetic change in the growing oak bud. It develops not into an acorn, but into something very different, a gall. Within, the tiny larva, whose secretions have caused the change, feeds on the oak tree's tissues. As summer proceeds, the galls become increasingly hard and woody. Autumn comes, and the oak tree starts to shed its leaves. It's shutting down for the winter. And with its leaves go both acorns and galls. Plant and insect life is suspended. But unseen changes are nevertheless taking place. Spring comes at last. Inside the gall, something starts moving. The larva has turned into an adult wasp. It has spent 9 months within the oak tree's tissues. It has only a few weeks of its life left. Now, as an adult, it must look for another oak to inject with eggs. A single oak tree may be afflicted by 70 different kinds of gall, each produced by a different species of wasp, and each with its own particular contorted shape. These hard shells may seem to be effective defenses for the little grub inside them, but not necessarily so. This is another kind of gall wasp, and she's not a genetic engineer, she's a burglar. Behind her, she trails her equipment for breaking and entering, a drill. She carefully selects a site for her operations and takes aim. She flicks away the drill's sheath and starts work. Her aim has to be very accurate if she's to strike her target, the larva at the gall center. The tip of her drill has a sharp cutting edge of metallic zinc, which pierces the gall tissues with ease. When she detects that she's reached the central chamber, a microscopic egg travels down the center of the drill and into the larva. The operation is over. Her offspring will now hatch in the gall's center, consume the flesh of the resident larva, and take over the gall.
Hey everybody, stop clicking around. I am in the woods with some water going in the background near my house.
Oh, that didn't go for the full 5 minutes. Oops. Okay, well, I'll tell you about, we can actually watch, hang on. Okay, we're still all together. Well, I'll find the rest of it because there is a whole one somewhere there. And I'm sorry if I sent you the wrong link. It's all right. All right, we'll catch up, I promise. Okay, so next time, look for the next installment. And we also have it up, Kim already has it up on her website, right? So you could go to the website and find it. Okay, but you did see the alternate generations, yay! And you saw one parasitoid, and we'll see the next time, we'll see what. So that's good because I'm trying to find lots of things to, it's awful to say, but to try to entertain you, because I don't want Joyce to end up in such a hurry that she cuts off all her fingers when she's dissecting. So we're going to give her time to set up each dissection. She's going to do it in real time, but we want to give her time without losing all the bits of her fingers. So that's good because we'll have something to present tomorrow. Now, oh dear, and I've got to finish, right? So now I will finish. We will share screen, and I'll share screen with the last one, which is here. Okay, so this, starting at the wrong end, and we're gonna go, this is for tomorrow. Slideshow, I'm sorry, I just did this there. Okay, this is what we're gonna be doing tomorrow. And why, yes, I did show you a few dissections, and I will show you lots more, and Joyce will be doing them in real time, which is very exciting. And it's going to be a surprise. Whatever happens tomorrow, we're going to get a lot of surprises, and that'll be fun. So, and the fun thing is, we looked last time, She had some inculins wandering around, and I might have some gall inducers wandering around if I'm lucky. And if not, I'll cut them open and they will wander around. So we'll see some live stuff. That'll be fun too. Anyway, when we dissect galls, we can see various tissue layers or lack thereof. So we can see how complex the gall is. We can understand about the feeding larvae, where they were and how it worked. And we can also see a history of how the gall was formed. This gives us a better idea about their biology, which is why we're doing it as good biologists. Okay, so here is, we've seen this before, this beautiful fuzzy pom-pom gall. It's on saltbush. And here we go, or was that, anyway, or coyote bush. This is a single midge larva in the middle of the gall tissue. So that's what we can see inside. That's a sessidemyiid. Here, and this is interesting, I had to look twice to see it. This is the gall. This is on a leaf, but it's made a leaf roll. So this is a Contraria species on this Spirea plant. And here we see several, not one, but several bright orange midge larvae wandering around inside the gall. So thanks to Joyce for all these wonderful dissections and photographs. Here is Androcus crystallinus. And with a video that we may see tomorrow, and you can see on your own anytime you want, the deep look video, this was what attracted the crew to actually let us do that film. I had suggested it when I first met them, but they went up to Mount Diablo with the idea of filming tarantulas. And they looked at the oak trees and they came back because that was a year when all of the oak trees were covered with this gall. They came back and they said, the oak trees looked almost red with the gall, and we want you and Joyce to come help us and make a video. So we did. All right, so this is crystallinus and it looks incredible. This is a really great, there's actually several separate little chambers on each one, as you'll see. And Joyce has taken a single chamber here and dissected it. And here we see the overwintering, waiting for spring, adult wasp. Whoops. Okay, here's Androcus parmula. And I saw a lot of fresh ones at Joyce's camera setup just a few days ago. So I'm very sure we will get to see these dissected in real time tomorrow, or if not, you know what they'll look like. Here is a little gall wasp trying to come out. Now she's a female, and while we don't know for sure, I can tell she's a female, and I know because they are all female. But look, she doesn't have a notch, okay? How I would know from the antenna, I don't know if you can see my pointer, it's straight, it's not notched. And this is slightly clubbed. It's a little bit larger at the end instead of being all the same size. That's little hints. And when she comes out, you'll see a ovipositor and know for a fact she's female. All right, here is Amphibolus quercus palmiformis on agrifolia. Actually, this is, coast live oak. This is, it looks like a very old gall, but this is what it looked like inside. So there's nothing alive and fresh here. This was from some years back, and it might have had points and they've all smoothed out over time. But you can see how many separate, let's see if I get my pointer back. You get 1, 2, 3, 4, 5, 6 separate chambers. And in each case, the larva has tunneled out, each single larva has tunneled out in a different direction and made a hole, an exit hole at the surface. Now we know that the larva probably actually chewed it while it was still larval, before it pupated, chewed it all the way to the edge, and all the wasp is going to have to do is push the edge out, and then she'll be free to fly away. So, but they tunneled to the edge. Here is Discocaspis simulata. This is a beautiful fuzzy gall. And I think that we'll get that clear whether this is a white oak or not. I'm not sure. But here is the chamber, and the wasp is inside of this chamber. We didn't cut this open, but we just showed you. There's always a separate tissue layer of a central chamber for cynipid galls. Then there's this tissue layer, and there may be other tissue layers in other galls. And then you see the outside tissue layer with the fuzzy. All of this is a typical gall. Now, these are acorn galls, Caloritis flora, and this is also on live oak, post live oak. And here we see all of the larvae. So this is going to be a polythalmus gall, lots of larvae inside a single acorn gall. And there you go. Now this is just a gall that's been, okay. This is Androcus vaccinifolia, but we have a lot like this. This is the classic, quote, oak apple. Not our California gall, but which is sort of solid and looks like a green apple on the outside. But the classic oak apple has a central chamber and lots of tissue, thread-like tissue, holding that central chamber in the middle, so that there's papery, thin rind on the outside, and then little spindly tissue leading to a central chamber. Now, if we cut that open, we would find a gall, probably a larva, because this looks pretty early in its life cycle. And here is the one I promise you I'll get more of and active. Okay, this is an Androcus kingi, probably cut open just about this time of year. So most of them are already mature adults, and if you put a little light on them, they'll be totally confused and think it's time to wake up, and they won't even be able to lay their eggs in any kind of a bud, because there won't be any buds on the trees. Oh well, poor baby. So we won't do too much of that. We'll just cut a few open for science. Okay, so between now and tomorrow, you may collect galls if that's permissible, or take photos of galls, or bring your galls, or bring your photos of galls. And I'm wanting you to use the provided. Joyce has made us a beautiful gall field guide to California galls. So see if you can use that for a lot of the galls you find. Don't have to, but if you try it, we'll see how well that works. Otherwise, I mean, you could, even if you've identified it, bring it in. We're happy to look at the gall, and this will be show and tell. And we also have collected a number of galls already. So I'll be doing photos and videoing my galls. And Joyce will actually do her dissection in real time. And we'll be happy to do your identification questions and my videos while Joyce sets up her dissections. And hopefully we'll save all 10 fingertips. But that's the plan. So we'll get away from sharing and open it to questions. We're only a little over time, I think. Sorry for those of you who are planning to be gone. Go ahead and go. We'll see you tomorrow, maybe.
Yeah, if you have any questions, please unmute yourself and you can ask it. In the meantime, I just wanted to share with you all where you can go to see more of this info. So I shared to my people, and I believe Emily sent to you guys this little Padlet that we have set up for our docents. And if you want to look at that deep look video, it's this one right here that says Deep Look in Oak Galls. The one that we watched with Attenborough is here, and the link to the insect book that we mentioned is also up here, as well as the oak talks that were done by Kate Marienchild and Paul. So lots of resources there for you. Does anybody have anything that they'd like to ask? Or
I have one question. Is it possible for a gall to thrive on dead wood?
If there is no, actually, no. If you find something that's still growing and the wood is dead, it's not a gall.
Oh.
So galls only, because the nutritive tissue they get for the insect to eat or mite, it has to come from living tissue. So if the wood is dead, no.
Thank you.
Sorry.
Do the parasitoids recognize, or do they parasitize alternate generations, and do they recognize the alternate generations?
You know, that is a good question. And I have seen fewer parasitoids from spring galls, and spring galls are a lot harder to find and a lot shorter of a generation, but they're out there. Parasitoids are out there. So I'm not sure what all is going on with that. That's a very good question. How good they are as far as alternate generations. Some of my European colleagues have done better work on that, and I will check their work and look up some of that and ask some of them. Might get a better answer from them. So, yeah.
So that kind of suggests the question: do the parasitoids have alternate generations, or are they a longer-lived species? Or
no, okay.
Sorry, I didn't.
No, they're fascinating. And in fact, there's a whole, you know, Joyce has a whole poster, which we'll show you next time. I'll have her, yeah, I'll have her send the rest here. So one on just parasitoids and stuff in wasps, so the wasps on the galls. But yeah, we've got lots of parasitoids, but there are whole species of parasitoids that are not all that fussy, that you'll find on lots of different host insects. I'm not sure how they manage it, because there'll be different sizes depending on the size of the host, which is really strange. But that's rare. Mostly parasitoids have to, okay, parasitoid wasps, and this is really interesting, I think, almost all insert some kind of virus. In the case of cynipoid, these are the fidgetids, and well, this is a group we haven't talked about, but the close relatives of cynipid wasps are parasitoids, and they actually have a virus-like particle that they insert at the time of oviposition. Apparently, the virus can actually calm the larva, the host larva that just has to wait to die, and you know, it has to be calm to wait to die to get eaten alive, we think. But now in the case, oh, this is so embarrassing. All right, I hope we have time for this. I'm sorry if we don't. Is it okay, Kim, if I go on for? It's going to take a minute or so more. Okay.
It's okay, and anybody who needs to leave will not offend anyone by dropping out.
Right. I'm not offended, I promise. And I hope to see you tomorrow, but I won't be offended if I don't. But anyway, the parasitoids, these are hyperparasitoids. So the ones that I'm studying, the fidgetids, are inside. They're endoparasitoids of the wasps that are endoparasitoids of aphids. So the very first thing in the key is it has to be less than 1 millimeter long, or it can't be this. Okay, now what do aphids do? The parasitoid actually has given some kind of a virus, we don't know, and also some other chemicals that actually controls the aphid's mind and behavior. So the aphid that is actually parasitized crawls, instead of on the stem where they normally hang out drinking plant juices, crawls over to a leaf, usually on the underside of a leaf, where it's not so obvious, and glues its tummy, or the front of it, to the leaf, and forms what we call an aphid mummy. And yes, it's almost Halloween, and it's really an aphid mummy. Okay, so this aphid mummy is this poor dying aphid being slowly eaten alive either by the larva, the original parasitic larva, or that parasitoid larva is being eaten by the vegetated larva that I studied. So interesting, right? It has parasites on parasites. And yes, it is very odd behavior. And if you see an aphid mummy, what you see is this thing that looks very strange. It doesn't really look like an aphid, and it's definitely oddly colored. And when the color goes really off, I know it has a hyperparasitoid in it. Okay, Eddie. Okay, now I've really gotten far afield. Sorry about that.
No, thank you very much. That was great.
Okay, we'll have more of this, and maybe I'll even give you some more links, okay? And I've got some great links to parasitoids and mind control. Can you imagine? You know, they're actually controlling, okay, we've got some parasitoids that make ants crawl up to the top of a stem of grass and sit out all night. Now, ants are normally only active during the day, and they go back in their colony and behave like, you know, sleep at night. But no, this parasitoid, it's a liver fluke. It's a worm. One worm gets in its brain, or with ganglion, you know, with cerebral ganglion that passes for a brain in this poor ant. And the ant sits there waiting to be eaten by a passing cow. And the fluke will become a liver fluke in the cow. So parasitoids can control behavior, and many of them do. And many of them use viruses. Almost all of them use viruses. Everything we've really studied uses viruses or virus-like particles like mine do. So.
Very cool. Very cool. Thank you.
I didn't want to worry you about this. Yeah, it has nothing to do with our pandemic virus. That's not a mind control virus, trust me.
As far as we know.
No, actually, yeah, okay, all right. I will have to say, yes, I don't know everything about that, but there really are mind controlling viruses that control the minds of poor little arthropods, some of whom would much rather be doing something else. So yes. And there are actually parasites that control our minds, but we'll not talk about that tonight. They're not insects, and I'm not worried. Talk about that more when it's closer to Halloween, maybe.
Great. Any other questions, comments, anything else from anyone? Well, I think that was fabulous. I am so excited about tomorrow, and I can see that everybody else is really digging this too. So thank you so much for your generosity with your time, Kathy, and thanks to all of our amazing docents who came and spent some really fun time with us. So we'll see everybody tomorrow at 4:30.
Before we go, I want to say I really miss being able to do this in person. Joyce and I have done this kind of thing in person and had great times, okay? And then we'd have you all dissecting at once, and we'd have to run around from microscope to microscope and find it all. So maybe this is better in some ways because we can see exactly what Joyce is seeing and what she's dissecting. But I also normally bring a pile of books, which I plan to show you tomorrow. So I'm going to show you books. I'm also going to show you Joyce's posters, and I'm going to show you my insect drawer, my museum drawer full of galls. So you can see galls from California, some of the things that you could be finding or you may have already seen. So, now, museum drawers aren't as good because galls often get dry and they turn brown, you know, and the leaves turn brown. So it's not as exciting as finding them fresh and new. The way photographs are more fun and better because they last longer. But again, I'll show you what I can, and all the you know, hands-on thing, you can't put your hands on them, but I'll do my best to magnify them so you can see them. while Joyce is busy preparing dissections, which are going to be so exciting. I can't wait to see what she's going to find, because what we were looking at just 2 days ago, it was fascinating. Plus, there were some inquilins running around, and you could actually identify them and see all the details on them in her little collection there. So I'm really looking forward to tomorrow. It's going to be full of wonderful surprises.
Good.
Thank you so much.
Yeah.
Thank you. Very, very good.
Thank you, Kathy.
Thank you for being a good audience. Thank you for your great questions and your patience.
Good night, everybody.
Okay, good night.
Right.
Good night.
All right. Well, I thought that went really well, Kathy. Thank you so much. And
right.
just let me know what else you need for tomorrow. Any links
I think
or
we
whatever.
should be fine. Yeah. And thank you for what you did on your end. I really appreciate it.
No problem. Easy peasy. All right. Have a great sleep, and we'll see you tomorrow.
Okay. Good night.
Wonderful presentation. Thanks so much.
Thank you.
Thanks, Lori.